So if we want to understand the phenotype of a plant, and that is to say how the plant grows, how well it resists disease, or how well it tolerates stress, we need to understand what's called the central dogma of biology. So this process starts with the genetic material of the plant, or the DNA. So DNA is converted to a messenger molecule called RNA through a process called transcription. And then this messenger molecule is further converted into a protein. So proteins are sort of like the building blocks of the cell. They work together, combine, and sort of form this phenotype effect. So me, myself, I primarily study transcription, or the conversion of DNA into this messenger molecule, because I find it to be the most informative. It's the most fluid process in that gene expression is constantly changing within the cell. So when plants experience different conditions, different environmental conditions, different pathogens, different stresses, specific genes are being activated in that their expression is being turned on, whereas other genes are being repressed, whereas their expression is being turned off. So by understanding sort of the interplay between which genes are being turned on and which are being turned off, it gives us a ton of information and better allows us to understand the phenotype of the plant. So for the example of heads up, essentially what I want to do is when you treat a seed with heads up, what specific genes are being turned on and how do these genes work together to promote this enhanced plant defense that Colin's talking about. So like Colin mentioned, the company's been around for a long time now, about 30 years. So we have years and years and decades of field trial data. We know that it works in the field, but when they brought me on, I wanted to do things a little bit differently. So since I've been here, I've developed a molecular lab at our location here in Saskatoon, as well as a controlled indoor growth facility where I can perform smaller scale experiments. So within this controlled growth facility, as well as out in the field, I collect tissue. That can be leaf tissue, root tissue. I bring this tissue back to the lab. So once in the lab, I can extract the specific messenger molecule, RNA, from this tissue. And from this messenger molecule, I can quantify gene expression. So for each specific gene, I'm able to understand and extrapolate how its expression changes over time under different treatments, and sort of put all the puzzle pieces together and better understand the phenotype of that plant. Go ahead, Paul. So if we take the idea about the central dogma into plant defense, essentially in plants there are two main defense hormones. There's salicylic acid as well as jasmonic acid. And what these two hormones do is that they activate the expression of specific genes that are involved in plant defense. They also act as signaling molecules, where when this molecule is produced, it signals to other cells as well as other plants that they also need to activate their gene expression because an infection is incoming. So what we found with Heads Up is that Heads Up treatment specifically activates this salicylic acid hormone. So if we look at the graph on the right here, what we have is the specific expression of a gene that is activated by salicylic acid. So for this example, when the concentration of salicylic acid is high in the cell, the expression of this gene is going to be high. And conversely, when the concentration of salicylic acid is low, the expression of this specific gene is low. And like I mentioned, salicylic acid is activated when a plant is under infection. So what we see in the bars on the left-hand side here, when you take soybean seedlings that haven't been infected yet, you see the expression of this specific gene is quite low. The dark blue bar is the Heads Up treated soybean here. And what you can see is that it's slightly elevated compared to the control. It's about a 2-fold increase. However, when you infect these plants, and this experiment specifically infected them with this Rhizoctonia root rot pathogen, you see a much different story with the gene expression. You see that in particular with the Heads Up treated plant, you see an explosion of gene expression, about a 31-fold increase in the expression of this specific gene. And this sort of has to do with the sort of systemic acquired resistance response that Colin was talking about. So this SAR effect essentially allows for that Heads Up treated plant to respond quicker and a lot stronger compared to untreated plant here. So click the hit button. So right here is sort of the phenotypes of these plants that I collected the tissue from. So on the left, your untreated plant, and on the right, the Heads Up treated plant. So this root rot pathogen was really able to do a lot of damage to the untreated plant here, whereas with the Heads Up treated plant, you see less severe symptoms, and this is strongly correlated to the elevated expression of this specific gene. So I performed this exact same experiment, not only on this Rhizoctonia pathogen, but also on white mold. So like Colin mentioned, white mold is a late season disease where the plants are a lot more mature and it's a lot further along compared to this root rot. And we saw this exact same effect where you see the same spike in gene expression as soon as you induce an infection. I also observed this exact same effect out in the field. In 2024, I went to many different field sites across Canada and the US, collected tissue and brought that back to the lab. In sites where disease didn't appear to be present, we saw that small bump in gene expression like you see on the uninfected bar here, whereas in sites where we did see some signs of disease, we saw a much bigger difference like the bar on the right here. So this sort of illustrates that systemic acquired resistance effect not only happens in the lab in controlled conditions, but also out in the field. And that's what Colin and his father have been seeing for the last 30 years. So with that, I'd like to stop and ask if anyone has any questions. I'd be happy to clear anything up if anything was a little confusing, but if not, I'll pass it back to Colin.
So Nick, here's a question for you. You're looking at gene expression with heads up, and we're eliciting and turning on the specific gene related to plant defense.
Mhm.
And you've looked at this after 14 days after planting on the chart, but you've also looked at it further out than 14 days as well.
Yeah, so like I mentioned, we saw the same effect with white mold. And with white mold in the field, generally white mold only affects when the plants are flowering or shortly thereafter. So for that experiment in the lab, I grew plants up for 45 days till the point where they were flowering, and then I infected them.
Yeah.
Prior to infection, I saw the same sort of effect as the bars on the left, and after infection, I saw the same effect as the bars on the right here, where you see that jump in gene expression. With the lab treatments, I also saw a significant reduction in disease, similar to the pictures below there.
Yeah. And in a field setting, you've actually been out last season and taken tissue samples in September after disease has already set in, and you can still detect higher gene expression with heads up. Correct?
Yeah. So generally, if you sort of hit it right on the head, right when the disease is just starting to set in, that's when you're going to see the biggest jump in gene expression. However, the effect continues as the season progresses and as the disease progresses, in that the untreated or base seed treatment plant, which is the other check that we sometimes use, those plants aren't able to access the same sort of magnitude of gene expression as the heads up plant is. So even if they're able to catch up a little bit and have maybe half as much gene expression, the fact that there's such a big initial gap sort of, you can really see it out in the field as well.